In this lecture, we're going to take a look at the fundamentals of bioelectrical signaling. And bioelectrical signaling is essentially a type of electrical energy transmission. And so when we take a look at two different types of electrical based systems, the first is biological and the other is a non-living conductor here on the right hand side, I'll get to in a moment. But in terms of biological electrical based systems, the fundamental component of cells are cell membranes. And the cell membranes are those that generate electrical energy for communication. So, in between different systems, such as the nervous system and the skeletal muscle system, um, and the electrical energy generated by cell membranes is responsible for producing these communications. And the fundamental mechanism for bioelectrical uh, events comes from the movement of charged atoms or ions. And when those ions move from inside the cell to the outside or the outside to the inside of the cell, it changes the electrical environment on both sides of the membrane. And that's the key event for how neurons communicate inside the neuron, and that will eventually lead to communication between neurons or between a neuron and another cell. And to contrast that process with non-living conductors carry out the exact same function. Non-living conductors generates electrical energy for inter-system communications, and the non-living conductor does not move whole atoms. So for the cell membrane, the membranes are moving whole atoms, and those atoms happen to have lost an electron or gained an electron, depending upon the particular atom. Those are what ions are. On the right-hand side, here we see some electrical insulation exposing copper wiring. And the copper is responsible for those electrical impulses. And to get a better understanding of that, we take a look at the left-hand side in the orbital model of an atom, or also called the Bohr-Rutherford model of the atom, in this very simplified drawing where we have a nucleus of the atom with all of these circulating electrons that exist in different orbits or so-called energy shells. And electrons inherently have been labeled to have a negative electrical charge. Protons found inside the nucleus have a positive charge, while the neutrons inside the nucleus, being neutral, do not have an electrical charge. This is a simplified Bohr-Rutherford model of a copper atom. And as we can see here with the copper atom is number 29 on the periodic table because it has 29 protons and it has 35 neutrons. And we see that it has a series of electrons in these outer so-called shells. Now, in a neutral copper atom, it will have the same number of electrons as it does protons. In the neutral copper atom, it does not have a net electrical charge. But if we take a look at the location of these electrons, here we see this one sitting by itself in the sole electron in this outermost energy shell. And because of that, this individual electron does not have a lot of motivation, if you will, to stay inside this orbiting shell. As a result, the copper atom has a tendency to lose this outermost electron. And when the copper atom loses that one electrical charge, it now has it's been said to be ionized. And when you take a series of these copper atoms beside each other and in what you would find in a copper wire, we see a continual transfer of electrons between copper atoms, and that's what makes it a very good electrical conductor in a non-living system. But remember, in a living system, biological system, does not move electrons in and out of the membrane, in and out of, or through the membrane, in and out of the cell. It moves whole atoms that happen to be ionized. So in terms of the mechanism for the bioelectrical signaling, the mechanism for ion movement in and out of the cell occurs through what are called gated channels. And these are proteins embedded in the membrane of the cell, and those proteins have the ability to open a pore in the middle of the protein when signaled. So the protein itself is said to have a gate. And the question here is, what opens the gate? And this is a mechanism of facilitated transport, where the protein facilitates the movement of an ion into the cell or an ion out of the cell. And what is needed for that is simply a concentration gradient. So when the protein opens up, that ion will move from a region of higher concentration to lower concentration. So the protein contains portions that are referred to as gates, and gates generally have two general states, open or closed. If the gate is closed, ions will not move through the protein and will stay confined inside the cell or outside the cell. If the gate becomes opened, then ions will flow from higher concentration to lower concentration, either into the cell or out of the cell. And there are three basic types of ion channels based on the gating mechanism. The first type are ion channels that are said to be voltage gated. And so the question here is, what opens up the gate of that protein to allow an ion to flow, either into the cell or out of the cell, depending upon the particular ion? The electrical environment. If the electrical environment of the inside of the cell to the outside of the cell changes, that is very often a mechanism to open up a gate or to close a gate. Those are referred to as open or closed states. So those particular proteins that are sensitive to that electrical environment will change its shape based upon that electrical environment and will either open up a gate or will close a gate. Ligand-gated ion channels are not sensitive to the electrical environment, but respond to a molecule. So a molecule called a ligand will form a chemical bond with that particular protein in the membrane, and that binding of that molecule, depending on what the molecule is, will either open that protein channel or will close the gate to that protein channel. So ligand-gated uh, ion channels are sensitive to neurotransmitters. So when we talk about the chemical synapse is what exists between one neuron and another neuron, or a neuron and a muscle cell. What is the mechanism for that communication? It's chemical in nature. So the first neuron will release a neurotransmitter, a chemical, a compound, and that chemical compound will form a chemical bond with the protein on the second cell, whether it's a neuron or a muscle cell. Some ion channels are mechanically gated. And what that basically means is that the application of force to the membrane is sufficient to open up those ion channels, and that will result in the generation of a bioelectrical signal. Mechanically gated ion channels are typically found in what are called stretch receptors, and those are sensory receptors found in our skin and found throughout other tissues in the body. But to open up those ion channels and to produce a bioelectrical signal, some external force has to be applied to that cell housing the protein channel in the membrane for that action potential for that bioelectrical signal to be generated. So this ends this video on the fundamentals of bioelectrical signaling.